Everyone, and thanks for your kind introductions, and thanks for all joining seminar in such a early morning. Today I will talk about new sample delivery systems combined with time reserved X-ray liquidographies. Uh, okay, or the time reserved X-ray solution scattering to explore the biomolecular reactions with the microgram sample quantities. Yeah, yeah. In my talks, I will overview the benefit and the limitation of the TRXL, especially for the irreversible reactions, and then I will introduce our new method, serial X-ray liquidographies, shortly SXL, and explain the details about it. And finally, I will demonstrate our successful applications in various biological reactions. So as you know that all living organisms undergo complex cellular processes to sustain their lives. And and these processes actually involve a series of chemical reactions and regulated by molecular machinery, mostly proteins. So to understand these functional laws and the molecular mechanisms, many biochemicals and structure study have been carried out, but however, these biomolecule in a solutions are not static. I mean that not not frozen inside parcels, and they constantly move and interact with other molecules and undergo a significant conformation change during biological processes. Hence, to fully understand these biological reactions, we need to the look at both the structures and the kinetics of these molecules. For this purpose, time-reserved X-ray liquidography, which is also called as a time-reserved X-ray solution scatterings, have been developed it, and it is a powerful technique for characterize molecular actions under physiological conditions. Yeah, thanks to the established setups and the sample delivery method, we we haven't used TRXL to study such a reversible photoreactions such as those involving the hemoglobin or myoglobin and PRPs. Despite these advantages, conventional TRXL setups can face challenges with irreversible reactions. Actually, these many biomolecules exhibit irreversible properties, and they are non-photoactive. To explore these reaction dynamics of them, then we have to use photocages molecules throughout the indirect triggering method. And considering just typical parameters to study these reactions, just 0.5 millimolar sample concentrations and molecular weight about 40 kilodaltons, and maybe we use a 10 hertz repetition rate with a 60 shot accumulations, and then the required sample quantity for data collections goes up to the 3.6 mLs of sample per hour, and this is equivalent to about 70 milligrams per hour. Actually, we know that this experiment in this beamline lasts a day or more than weeks, so the required sample quantities goes up to the impractical numbers. Consequently, TRXL have been limited to investigate the reaction dynamics of reversible photoreactions only. So imagine these situations: a biochemist from other groups, or just a young colleague in your labs, and come to you and say that he he or she got new structures and wanna study its reaction dynamics with trial Excel. Then maybe you can say that, yeah, if your proteins are poroactive and undergo reversible reactions, just to provide me a few milligrams of sample, then I can give you some kind of data. But if the reaction is not reversible and irreversible, what I mean, or the non-poroactive, then you have to say that, hey, bring me 100 milligrams of sample, only just for the pilot experiment. And if you say that, then the colleague, what 100 milligrams, and they span several months for such a preparations. And then this promising proposed work will be challenging and likely to be overlooked and just left in a shelf, and we forgot about the project at all anymore, and we did not care about it because it requires a lot of sample quantity. It's really hard to prepare. So you think this scenario seems to be a little bit quite distorted, right? But yeah, as a matter of fact, this young guy was me and my teams, and I spent several months to prepare such a large quantity of sample for a single visit to the beamline. Yeah, later I recall that over I we have dedicated over 50% of our research time and substantial lab resource solely to the sample preparations. And these realizations actually pushed me to look for a better way, a better solution leading to the development of a new sample delivery systems. So, right, let's start it over from capillary setups. Yeah. This schematic actually show simplified schema of the process in TRXL. As you can see in a detailed, here, as you can see the detailed size view of the capillary, then you will realize that on most of the sample quantity are wasted only for maintaining the sample delivery systems. And considering a tiny size of the X-ray compared to the laser, only about 1% of sample is just used for the data collections. So actually, now we handle the irreversible reactions. So the used sample portions cannot be recycled. Actually, that is a mixture of the reactants and products. So here comes my theory. If we can array these tiny sample portions in a overlapping region between the X-ray and lasers in some area in a space, not in a capillary, then we can collect a equivalent data set from a 1% of the sample that used in a paper. That is about 36 microliters per hour. And in other words, it's less than 1 milligram. Now maybe young biochemists will happy to make it, I think. So when design these reaction chambers, we consider these parameters. First, we to enhance the data collection efficiency, so we array the nanoliter sized microchambers, not a single way, two-dimensional way, and second, to load the liquid sample easily, and then keep the scattering signal intact on these truncated scale pyramidal shape is used. And third, to reduce the sample uses further, we rotate the reaction chambers in a 45 angle while we keeping the tolerance in goniometers movement. And last, we designed this chip in a asymmetric layout, which one side is widely opened for the sample loading and the other side is closed with a PDMS material to prevent such rinkings and some leakings and unexpected things happen. So finally, as a proof of concepts, we made a 40 by 30 array totaling 1,200 micro chambers, and it's with volumes about 4 nanoliters. Notably, this reaction chamber is totally isolated to ensure that the reaction occurs in a controlled environment and not mix each others between the collections. So I would like to mention that when we made the SXA chips, and then we used the PDMS material combined with organosilicon detergent to improve the hydrophilicities of the chips. So this chip have a high hydrophilicities, and so easily the sample can be easily loaded onto the chips, and after loading, the chip is sealed it with a thin polypropylene films on a open side, and the assembled chip keep the samples per more than several hours, which is sufficient for the data collections. So this slide covered the sample loading scheme, sample loading photographs and the binline setups for SXL experiment. And SXL experiment was performed at biocartin line, and the other parameters is almost similar to the typical setups. So to validate our SXS chips, which contain the thin films and PDMS backside in our X-ray pathways, which may affect the scattering signal. So we have to check the background scattering signal from each each component. So since you can see that the film part and the film with the chip part, and after loading the buffer, after proteins and lasers, you can clearly see uh, uh this part is clean enough to clean and make it to extract the protein signal from scattering, and also and also you can we collect a scattering signal from multiple microchambers, and you can see that scattering signal and the different scattering signal is almost identical, and 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 this uniformity confirm that our SXF chips work and are suitable for the applications. So next we apply the SXF method to study irreversible reactions, and we focus actually we chose the UVR8 photoreceptors in plant stress responses triggered by harmful UV light. And based on the previous studies, and UVR8 undergoes an irreversible dissociations from homodimer to the monomer, often UV illuminations. And to trigger the reactions, we use the 300 nanometers pump lasers, and we used about 38 micrograms in totaling for the chips in the case of lower concentrated the sample. And we used about 115 microgram for high concentrated sample that is on the middle of the figures. And as you can see that the time reserved signal becomes stronger during the reactions, indicating that this dimeric UVA undergoes a significant structure change, and from tens of microseconds to the several milliseconds. And if we increase the sample concentrations report, then we can clearly see the pictures, and this high concentrate sample enhance the signal to noise ratios while we retaining the similar kinetics result. And for the detailed comparisons, let's bring the previous single shot scattering result. For SXL, each reaction chamber required about 31.6 nanograms, and for the high concentrated one, it's about 100 nanograms for the each pump probe measurement, and we used about 1.3 micrograms for the different scattering curve, which is come from 40 shot accumulations. So for the comparisons, we also used the closed capillary setups, and we spent about 370 micrograms for sample loadings. And after a single measurement, you can clearly see that the different scattering signal diminish rapidly right after single punch, because it is irreversible. So due to the irreversible natures, then you can compare the scattering signal from the SXL and the capillary setups, and SXL just use about 4 micrograms of sample per the time reserved data, while the conventional setups consume about more than 300 micrograms. So it means that SXLs have a great sample efficiency than the conventional setups. And for we test the irreversible reaction, and then we wonder that these irreversible reactions like dissociations, the time reserved signal is usually strong enough for the detections. But however, for the weaker signal from a local conformation chain, maybe SXR cannot detect it. So we chose the LOVE2 domain from Avina sativa phototropin, and this domain undergo a structure change in response to the blue light. So any if the SXL efficiently to check the efficiency of SXL to collect data from such a subtle change. And since this reaction is reversible, we better to collect the time reserved signal from conventional setups, and so that we can compare the time reserved signal obtained from both setups. So we used about 400 micrograms per the closed capillary setups. And as you can see that below, you can see the photograph below, then the process yellow color sample turn into actually dark green colors after few repeated measurement. And this color change actually indicated that the sample is severely damaged from both lasers and X-ray pulses. Consequently, we only obtain about whole reliable difference scattering curve from conventional setups. So that is about that is each curve consuming about 100 micrograms for the data collections. And in our XXL experiment, we used about 170 micrograms in total, and about 140 nanograms was used per the each pump probe measurement, and about 4.6 microgram per the 4.6 microgram is used per the different scattering of curve, and and as you can as I told you before that in a closed capillary setups we used about 400 microgram for the pore difference curve. So the your sample was about 100 microgram for each image, and we spent about 5.6 micrograms for the equivalent data set. So this finding actually indicated that even for the reversible photoreactions, when the sample is sensitive to the repeated measurement, then our method offer a significant advantage over conventional setups. Here is a result from a successful experiment using both wild type and mutant LAB2 domains, and the mutant domain exhibit a faster photo reactions compared to the wild types. So as you can see the photo cycles between them, and SXL effectively distinguish the difference in high SNR. And from this previous experiment, we didn't actually change any beamline setups between the experiment, and so we didn't waste any beam time for sample change, sample wash, or other things. So this reader suggesting that our methods can be used for the screening the reaction dynamics of the protein under various conditions as same platforms. Actually, that is limited, and we didn't do the job with our conventional method. Hence, we collect our time reserved data from various conditions such as rate pulses and ionic strengths and various pH condition, even under chemical denaturant conditions. And as you can see, the reaction progress from each run, and then SXL can co- easily produce data set to be distinguished. And then you can see the distinct features between the data collections. And finally, we apply the SXL to the non-polar active molecules, and we conduct a pH jump experiment by photolyzing a photocated sulfate with lysozyme. For these demonstrations, we used about 300 micrograms of lysozyme and 13 micrograms of photocages. Uh, we successfully collect time reserved data from lysozyme, both with and without photocages. And as you can see that the results clearly show that the only lysozyme in the presence of photocage undergo a significant conformational change. And in summary, our sample delivery systems combined with the TRXL effectively address the sample quantities issues of conventional methods. And our proof of concept study demonstrate that the SSL can successfully handle both irreversible and reversible reactions with just a microgram level sample quantity. And looking ahead, actually, SXL show a great potential as a versatile platform to exploring diverse conditions or inhibitor screen and complex kinetic studies. Before closing my talks, I would like to express my sincere thanks to everyone who made this work possible. I special thanks to my team for their dedications and effort, and I also thanks to the BIM9 scientists for their invaluable support and feedbacks on this project. And finally, I appreciate all of you for your kind attention. Thank you for, thanks.